Hello everyone. I figured we'd talk a little bit more about SN2 in the context of biology since I know you guys are interested in biology type stuff. So what we're going to talk about is the role of SN2 in DNA, and this is related to making better leaving groups using phosphates. So we were talking today in class about using alcohols and turning them into better leaving groups, but we just used Br. But your body doesn't use that, it uses phosphates instead. So let's look at that. So before we do that, some of these drawings are pretty complicated, so it'd be hard to draw them like freehand. So I'm going to use slides, PowerPoint slides, and I wanted them to be available to you guys so you didn't have to draw it all by hand. You can either use it on your tablet or you can just make notes next to the slides if you don't want to print it, or you can print it out. But where they are is they're going to be in the supplemental. So if you look right, let me get the pointer. So if you look right here, this is in the supplemental. It's in the section 2 supplemental, since that's the section we're in, and it's going to be lecture 20. So if you look here, you can have either the PDF version or the PowerPoint version, whichever you prefer. The PDF version, I made it 4 slides per page just to save paper, but you can still zoom in, or you can use the PowerPoint one and make a different PDF if you prefer. So that's where you're going to find those. So if you want to download those and write on them, you can go ahead and pause the video and do that now, and then we'll move on. So first off, let's start with the big picture. So if we're looking at biologically important molecules, we see the structure here on the right. So what is this structure? Just looking at it. Right, it's DNA, right? We've all seen DNA before. So we know that DNA is a double helix, right? So we've got the double helix here, and we've got these different nucleic acids or nitrogenous bases, whatever you want to call them, thymine, adenine, cytosine, guanine. So it must be DNA, it's not RNA, right? But generally, if you ask someone what DNA is made up of, that's kind of the answer you're going to get, right? You're just going to get, there's these bases, and then they match up, and everything's great. Let's think a little bit more on a chemical level what DNA is made of. So like we mentioned before, we have these different bases, and they're sometimes called nucleic acids, they're sometimes called nitrogenous bases, but the structure is what we have on the right here. So this is the structure of adenine, this is thymine, this one's guanine, and this one is cytosine. It's labeled there, just kind of covered up for me. And so those are the 4 different bases, and you can see they've got all these different structures. They've also got hydrogen bonding, which is what's shown in between the bases. That's why you get matching and you can get the complementary strand because you have hydrogen bonding. So it kind of shows you that here, but we're not really going to focus on that today. That tends to be what most people see. What we want to focus on is what's going on with these purple strips. No one really talks about those. That's what's holding all the DNA together. So you can see over here on the right, it says we've got deoxyribose sugars, and that's part of the purple, and then there's these little lines of phosphates. We're going to talk about the phosphates and how they hold stuff together, okay? So if we take a closer look, this is the DNA strand of just one side, and this is the deoxyribose sugars and the phosphate thing that's holding everything together. So if you see, here's our bases, those are normal, but what it's attached to is this part's the sugar, which we'll see more about sugars at the very end of this class when we talk about carbohydrates. So we'll talk a little bit more about those structures. But these are the sugars, and then these are the phosphates. These are the POs that are holding everything together in the chain. But what we don't see is that actually the phosphates also have another role in putting these things together. So we're going to talk about that as well. But we do want to recognize the structure of a phosphate. It looks like this. You can see a little bit closer on this side. There's different ways you can see it drawn, but it's usually going to have P with 4 oxygens on it, and then sometimes they'll show double bond O, sometimes they won't. It depends on the structure. And this one has negative charges. It depends on the environment if it's going to have negative charges or have OHs here, but this book drew them all as negative charges. All right, so you can see this is the 5' end, this is the 3' end. That has to do with the carbohydrates. We're not going to focus on that, but we really do want to focus on how the structure is important and what the chemicals in it are. So that's what we're going to focus on. So since we're talking about structures of DNA and phosphates, I thought I would just mention that the main difference between a nucleoside and a nucleotide. So a nucleoside is the piece of the base that has the base and the sugar. So that would be in this molecule here, just the yellow part and the orange part. So it'd be like without the phosphate on here. So it would look like this. Then whenever you have a nucleotide, you're going to have the base, the sugar, and have a phosphate group. So here the structure would have a phosphate, so you can see that right here, I added that, and then that's going to be a nucleotide. So ties have phosphate, and I always think of like detergents have phosphates, which is something that maybe you didn't know, but Tide detergent, so I always think like, oh, that means it has phosphates. So they have that phosphate piece. So here they usually have one phosphate if it's going to attach and to make a chain, but they can have more than one, and it can be used for different functions. So if you look here, there's lots of different functions of nucleotides. They can be used for data storage. That's basically what DNA is, right? DNA is trying to record the information you need to make copies of RNA to make proteins. So it's just like stored instructions for making things. It can also be used as energy currency. So like ATP, that's what that is, right? And then you can use it for cellular communication. So if you've seen CAMP or ATP allosteric regulators, things like that are regulating using molecules with just one phosphate. That M means one phosphate and the tri means three. And then they can also be used as coenzyme catalysts. So they actually make reactions go faster. And you can see some more information on this website here. But so those are some of the functions of nucleotides. So here is all those pieces broken down into separate parts, just so you can see it more clearly. So on the left, we've got our sugars. That's the sugar part of DNA or RNA. And if it's DNA, you're going to have deoxyribose, and if it's RNA, you're going to have ribose. That's the difference. Deoxyribose is the D part of DNA. And the only difference is this hydrogen here versus an OH here. So deoxy means we took away an oxygen. That's why it's called that. These are the bases. Again, we've seen those before. And then these are the phosphates. So it's just an easier way to see them if you didn't see them before. So there are several types of reactions that have to do with RNA and DNA chains. One topic that we can talk about a little bit later is condensation with the phosphates. That's what's actually attaching these to the sugars and making this long chain. So that's considered like a condensation reaction. And we'll talk about that. But what we're going to talk about today is something different. We're going to talk about how you form the nitrogen to this carbon bond, because that does not look like an easy bond to form. Normally for a sugar, there'd be an O here, and there's clearly no O. So something's happening. So we're going to do a substitution, so an SN2, but we're going to do it with a pyrophosphate. So we're going to see what a pyrophosphate looks like. That's going to be our better leaving group that we're going to turn the alcohol into the better leaving group. All right, so let's look at that. All right, so first let's look at the big picture process. So we said this is going to be a substitution reaction. So what is going to happen is a normal sugar has an OH here on the carbon, the 1' carbon, and this is going to be an OH group, and then this has a nitrogen here. So we see we've got a good nucleophile here, but then we don't have a good leaving group over here. Or we have a good nucleophile here and not a good leaving group at all. Over here, there's no leaving group. So we've got a problem. How are we going to get these two together? We can see if something's going to form, and we're going to lose water, so technically that makes it a condensation reaction, but we are going to lose water overall. But this is not what's really happening. It's not like a nitrogen's just going to come here and attack here and this O is just going to fly away. That's just not going to work. So let's look at what the two-step process is that will make this reaction actually work in your body. So this slide is showing the very first step of how we're going to get the base onto our nucleotide. So the very first thing we want to do is form a pyrophosphate. The pyrophosphate is the group that's shown in yellow, so right here. It's going to come from an ATP molecule, which your body has lots of, and we're going to add it to the sugar. So remember, we've got a nucleotide has a sugar and a phosphate. That's already formed in this part. And then we need to add, on this oxygen, we need to add the nitrogen base. So we're going to put the pyrophosphate on there first to make a better leaving group. So when you do that, you're making, this is the actual pyrophosphate. It has to have this oxygen attached to this oxygen. So it's actually got one extra oxygen than what this has. And so that's going to be our leaving group. So this whole thing here is going to be our leaving group for the next reaction. Now down here they're showing this AMP, but that's just showing that it's a molecule, it's like a byproduct. So it's like the leftover part of this is like the leftover part of this. So we're not really worried about that. That's just going to sort of float away and be used for something else later. AMP, to transfer it to make more ATP is like a thing. You've got an enzyme for it. So the mechanism for this reaction is going to be attack this oxygen needs to form to the phosphorus, right? So we can show it like this, where we have an arrow going from the oxygen, that's going to be our nucleophile, to the phosphorus, that's going to be our electrophile. And then part of this ATP is going to come off. So the leaving group is going to be here where this bond breaks and goes to the oxygen to make a negative charge down here. Now, if you look, they've actually not shown all the steps. This came from your book, but they didn't show the step where you take off a hydrogen. So obviously there'd be another step if you come down here. And then we'd have the whole molecule, but it's just going to have this extra hydrogen here, right? And you're going to have a positive charge on the oxygen. And then something's going to come and remove this in an acid-base reaction to get up here where there's clearly no hydrogen there. Now, you don't have to show this because I'm not going to ask you to draw this mechanism on a test. So you are going to have to know how to go from here to here, like on a box question, but you're not going to ever have to draw the whole mechanism where you're drawing all this stuff and all this stuff. It's just too much to draw. So it's just giving you an idea. So they just skipped that step. They were being a little bit lazy, but there is a step here where you would remove that hydrogen, and that's how you stop at the pyrophosphate. So that's what's going to form. So that's the very first step. So that's the mechanism and the step, and this is the pyrophosphate. So this is like a molecule that's stable and can be formed. It's actually not like it happens instantaneously in your body. It's going to form this first and then form another thing. So on a box question, I would have it go just from here to here with using ATP. That would be the box question. Okay, so then let's see what happens once you make a pyrophosphate. What are we going to do with that? All right, so now that we've formed the pyrophosphate, we're going to go to the second step where we're going to get our actual substitution. So here we're going to be adding the nucleophile for the substitution to occur. And the nucleophile is going to be our nucleic acid / nitrogenous base, whichever you want to call it. So we're going to have our nitrogen attached to this carbon, and that was the key thing we wanted, right? So if you look, here's the key carbon that we want it to attach to. So we'll call that carbon number 1. And then over here, this is carbon number 1, right? So this is where the reaction is going to happen, and this is our leaving group, the pyrophosphate. So remember we said that pyrophosphate is a good leaving group because it has this O on it added to the phosphate part. And that's what makes it a good leaving group, because then you get this O minus and it's got resonance, and there's all sorts of good things about that. So it's got the negative charge that we like to have for a good leaving group. It's a weak base, so it's very stable, and it's got resonance, so that's good. So that's why it's a good leaving group. Pyrophosphates are good. So the SN2 reaction is going to look like this for the mechanism. This is going to be our nucleophile. So the nitrogen on the nitrogenous base, which all 4 of them have, right? Technically 5 for RNA, but for DNA, all 4 of them. And so that's going to be our nucleophile. This is going to be our electrophile because this is the thing with the leaving group. It's just instead of having a Br leaving group, now we've got a pyrophosphate. So it looks kind of funny, right? Now the whole thing's going to leave with the O because we're trying to make a nitrogen carbon bond. So remember that, nitrogen carbon bond. So that's what we're going to do. So the first step of the SN2 is going to be nucleophile attached to the thing, right? And then the leaving group's going to leave. And then that's all you have to do. That's the whole thing. So it's just one step to go from here to here. And it's just like what we did before for all of our other SN2s. It just looks complicated because we have all these oxygens everywhere, and the leaving group's big, and the nucleophile's big, and the thing with the substrate is also big. So it's just kind of a pain to draw, but it's not too bad. So again, I'm not going to ask you to draw the mechanism on a test, but I would ask you a box question. So I might say, like, if this is our thing with the pyrophosphate, then what's it going to look like when I add a nucleophile? I might add a nucleophile that's a base, or I might add a different nucleophile, right? I could add like a smaller nitrogen thing as opposed to this giant nitrogen thing. I might have a substrate with a sugar, or I might just have a substrate that has a pyrophosphate that's just like a regular carbon structure, just to make it more simplified. And you can look on the practice for today's activities, and you'll see examples that are a little bit more simplified organic versions, but the reactions are the same. So make sure that you try those and see how they apply, and then let me know if you have any questions, okay? All right, so then that's all we're going to talk about for the biology part for SN2s. Remember, just focus on the reactions. You don't have to worry as much about the structure part at the beginning. It's just all lead up to get to the reaction part, which is what we want to focus on. Hopefully you found this interesting, and it was a good application of the biology, and you can see how it relates to the alcohol stuff that we talked about in class today. If you're not sure, or if you're still having trouble with the activities, please let me know, and I'll be happy to help bridge that gap for you. All right, so hopefully it's helpful. Make sure you try the activities, and we'll see you next time. Bye.